When you hear the word cholesterol, what comes in your mind? Heart attack, blocked arteries, something bad. But what if I told you that without cholesterol you wouldn't even survive? Every cell in our body needs cholesterol. It's essential for making hormones, vitamin D, bile acids, and even maintaining healthy cells and membranes. So, why does it sometimes become deadly? In today's video, we will uncover the complete journey of cholesterol from how it's synthesized in liver, how it's transported through the blood by LDL and HDL, to how excess cholesterol leads to atherosclerosis and heart diseases. By the end of this video, cholesterol metabolism will feel like simple story, not confusing pathway. So let's start. Found cholesterol found exclusively in animals, hence called as the animal sterol, and total body content is 2 gram per kg. It is the amphipathic in nature, both hydrophobic and hydrophilic. Its functions. Cholesterol is essential to life. It is the structural component of the cell membrane. Cholesterol is a precursor for the synthesis of all other steroids. These include steroid hormones, vitamin D, and bile acids. It is essential ingredient in the structure of lipoproteins, in which form the lipids in the body are transported. Fatty acids are transported to the liver as cholesterol esters for oxidation. Cholesterol biosynthesis. Let's discuss. Introduction. 1 gram of cholesterol is synthesized per day in adults. Almost all the tissues of the body participate in the cholesterol synthesis. Let's see about the site and requirements for synthesis. Site is cytosol and smooth endoplasmic reticulum, and main organs are the liver, it is the major organ, and intestine, and then skin, adrenal cortex, and reproductive tissue. Major synthesis occurs in liver, it's about 20%, and then intestine 15%. Precursor is acetyl-CoA, and energy required in the form of ATP and NADPH. It is because of the reducing power. For 1 mole of cholesterol, 18 acetyl-CoA, 36 ATP, and 16 NADPH is required. And how is become cholesterol? Let's discuss about the stages of the cholesterol synthesis. There are the 5 stages. You should mug up these 5 stages. First is the synthesis of HMG-CoA, then formation of mevalonate, then production of isoprenoid units, then synthesis of squalene, and then conversion of squalene to cholesterol. You should remember cholesterol is 27 carbon compound. So from 6 carbon to 27 carbon, here is the step-by-step biosynthesis of the cholesterol from 6 carbons to then 5, then 30, and then reduction to 27. Let's see about the first reaction. Synthesis of beta hydroxy beta methyl butyryl CoA means the HMG-CoA. It is the 6 carbon compound. Acetyl-CoA plus acetyl-CoA on the condensation forms acetoacetyl-CoA. It's little bit related to, like similar to ketogenesis. In previous video I have told about that. Acetoacetyl-CoA plus acetyl-CoA then form HMG-CoA with the help of HMG-CoA synthesis. Similar reaction to that ketone body synthesis, since ketogenesis occurs in mitochondria and cholesterol synthesis occurs in cytosol. Only difference is there in these steps that the ketogenesis occurs in mitochondria and cholesterol synthesis occurs in cytosol. And HMG-CoA exists in 2 pools, like 2 sources of the cell, in the cytosol also and in the mitochondria also. Then second step is formation of mevalonate. It is HMG-CoA reductase enzyme, that is the rate limiting enzyme, and the site for this reaction is endoplasmic reticulum. HMG-CoA on the reduction form mevalonate, and here used 2 reducing equivalents, that is 2 NADPH are used and forms mevalonate. Third step is production of isoprenoid units. 3-step reaction. It is catalyzed by kinases, and ATP is required in these reactions, and Mg+2 is required. These are the 3 reactions. First, mevalonate is converted into 5-phosphomevalonate with the use of mevalonate kinase. Then 1 ATP is used. 5-phosphomevalonate converted into 5-pyrophosphomevalonate with the help of phosphomevalonate kinase. And Here you can remember as this name and their addition of kinase. This is the enzyme for their further reaction. Here 1-ATP is used. Then 5-pyrophosphomevalonate forms in 3-phospho-5-pyrophosphomevalonate. It is like big names like that, but you can remember after reading 2 to 3 times. And then used 1-ATP with the help of kinase enzyme and Mg+2 is required for these enzymes of reactions. 3 steps. First step, second step is decarboxylation, and third is isomerization. Here 3-phospho-5-pyrophosphomevalonate undergoes decarboxylation, means removal of CO2, and forms isopentenyl pyrophosphate. It is of 5 carbons. Here is the enzyme pyrophosphomevalonate decarboxylation. Isopentenyl pyrophosphate, it is of the 5 carbon, forms the isopentenyl pyrophosphate isomerase. It is the IPP, and it is forms dimethyl allyl pyrophosphate of the 5 carbons from the enzyme isopentenyl pyrophosphate isomerase. Its short form is DPP, and then DPP and IPP are both the 5 carbon isoprenoid units. Then fourth step is synthesis of squalene. It is the 30 carbon compound. How it's formed? Let's see. The IPP and DPP, these are the 2 isoprenoid units, condense and then form geranyl pyrophosphate, means the GPP of the 10 carbon. These 5 plus 5 becomes 10. Then GPP plus IPP on the condensation form farnesyl pyrophosphate, means FPP. It is a 15 carbon, 10 plus 5 15. And then finally both 15 units FPP plus FPP, means the farnesyl pyrophosphate, forms the squalene with the enzyme of the squalene synthesis. Finally 30 carbon compound is formed. FPP, besides its participation in cholesterol synthesis, it involved in also formation of the ubiquinone and then prenylation of proteins. Next, fifth step is conversion of squalene to cholesterol. Squalene is 30 carbon compound converted into lanosterol, that is also 30 carbon, but using enzymes using reactions hydroxylation and cyclization with the help of oxygen and NADPH units. Here cyclization is occur, then lanosterol is converted into cholesterol with the series of 19 enzymatic reactions. Here are the key changes that from lanosterol to cholesterol, that reduction of the carbon into 30 to 27, it is the major change. Then removal of 3 methyl groups, that's this 3CH3, hence 3 carbons removed. Then double bond shifts and penultimate product, that is the 7-dehydrocholesterol. Then reduction occurs and cholesterol is finally occurred. Then how regulated this process of the cholesterol synthesis? Cholesterol biosynthesis is controlled by the rate limiting enzyme, that is the HMG-CoA reductase, at the beginning of the pathway. HMG-CoA reductase is found in association with the ER and is subjected to different metabolic controls. First is feedback regulation. It is the end product control, that is the cholesterol itself inhibits its synthesis. How? Increased cholesterol decreases the HMG-CoA reductase synthesis by decreasing the transcription of gene that responsible for production of HMG-CoA reductase. Then the hormonal regulation. HMG-CoA reductase exists in 2 interconvertible forms, in the dephosphorylated form and phosphorylated form. Then glucagon and glucocorticoids favor the formation of inactive HMG-CoA reductase and decrease in cholesterol synthesis. And insulin and thyroxine favor the formation of active HMG-CoA reductase, that is in dephosphorylated form, increase the cholesterol synthesis. Inhibition by drugs, compactin and lovastatin, that is the fungal products used in decrease the serum cholesterol levels in case of hypercholesterolemia, and competitive inhibitors of the enzyme HMG-CoA reductase, 50 to 60% in decrease in the cholesterol levels, results in the normal cholesterol levels in the body. Then the HMG-CoA reductase activity inhibited by the bile acids. Fasting also reduces activity of the enzyme. Then we will see the degradation of the cholesterol. Introduction, that the steroid nucleus, that is the ring structure of the cholesterol, can't be broken down in the humans. Cholesterol is 50% is converted to bile acids and then excreted through the feces, and the precursor for the synthesis of steroid hormones, that is vitamin D and caprostenoid and the cholesterol. First is synthesis of the bile acids, that is the bile acids that amphipathic passes both polar and non-polar groups, emulsifying agents in the intestine and participate in digestion and absorption of the lipids. Then second, it is the pathway, and we will see about the theory. It is the enterohepatic circulation. What is that? The conjugated bile salts synthesized in liver accumulate in the gallbladder. They secreted into intestine 50 to 30 gram per day, act as emulsifying agents for the digestion, and large portion of bile acids reabsorbed and returned to the liver through the portal vein. Thus, bile salts are recycled and reused several times in a day. This is called as the enterohepatic circulation. 0.5 gram per day fecal excretion of the bile salts is the only route for the removal of the cholesterol from the body. It is the important point that only way to removal of the cholesterol from the body through feces, and 0.5 gram per day synthesized in liver to replace the bile salts. It is the cholesterol in the liver then form its 7-hydroxycholesterol. Then it's converted into cholic acid and dehydroxycholic acid, and then forms finally in the intestine these deoxycholic acid that forms that is the secondary bile acids, taro or glycodeoxycholic acids in the through intestinal bacteria forms lithocholic acid. With the reactions deconjugation and dehydroxylations are formed, and secondary bile acids exist as a sodium and potassium salts. Then we will see about the synthesis of the steroid hormones from the cholesterol. Cholesterol is the precursor for synthesis of all 5 classes of steroid hormones: glucocorticoids, mineralocorticoids, progestins, androgens, and estrogens. These are the 5 classes of the steroid hormones. It is the pathway for the synthesis of the steroid hormones. Synthesis of vitamin D, 7-dehydrocholesterol, and the intermediate in the synthesis of cholesterol. It is the penultimate substance, and it is converted to cholecalciferol, means the vitamin D3, by the ultraviolet rays in the skin. Then how transport of the cholesterol occurs with the reversed cholesterol transport. Cholesterol is present in plasma, lipoproteins in the two forms. Which two forms? About 70 to 75%. It is an esterified form with the long chain fatty acids, and about 25 to 30% free cholesterol. This form cholesterol readily exchanges between the lipoproteins and also with the cell membranes. Role of LCAT means the lecithin cholesterol acyl transferase. It is the enzyme HDL, and this enzyme responsible for the transport of elimination of the cholesterol from the body. It is the lipoprotein. It is more important in the transport of the cholesterol. Then we will see about the reverse cholesterol transport process, by which excess cholesterol from the peripheral tissues is transported back to the liver for the degradation and excretion. These are the steps. That is, first is cholesterol efflux. Peripheral tissues like macrophages release free cholesterol. It is taken up by the nascent HDL. And second one is esterification of LCAT, this enzyme that the lecithin cholesterol acyl transferase synthesized in the liver, and that act as a transfer agent for the fatty acid from the lecithin of the cholesterol and forms the cholesterol ester, and cholesterol ester moves into the core, that is HDL becomes mature spherical HDL. Role of ApoA1 protein of the HDL, that is ApoA1 activates this enzyme LCAT, essential for HDL function. And fourth step is transport to liver. HDL carries cholesterol esters to liver directly via HDL receptors, indirectly via transfers to other lipoproteins VLDL and LDL, and then the excretion through the liver converts cholesterol through the bile acids and excreted via bile into intestine. Here are the major sources of liver cholesterol and its utilization. Here is the liver, and liver has cholesterol pool about 1000 milligram. Here dietary cholesterol 500 and cholesterol synthesis 500 milligram per day, total 1000. And then lipoproteins excrete variable, and cholesterol loss in bile is 500 milligram, and bile salts and acids 250 gram per day. Here cholesterol metabolism is over. We have seen the synthesis, degradation, transport of the cholesterol. These steps you should practice 2 to 3 times. You can remember. And thank you for watching. If you like it, share, like, and subscribe. Thank you.